Then I would like to switch to the human setting, because you also discuss, or it's not clear to me, do human macrophages actually generate polyamines?
That's a good question, and actually neutrophils in humans are supposed to have arginase 1, which was recently said in some Nature paper, but about macrophages we are not sure. But anyway, we were interested in arginase 1 and asthma, so with our model we if arginase 1 has some effect on either tumor or asthma, then it would be more interesting. Then maybe we can narrow our vision to what cells in human do express arginase and what is the effect. Because with our experimental model, we deleted arginase from all the myeloids or all cells in the lung, and we don't have any effect. If at all we had seen an effect, we would be more curious to know how to mimic it in the humans. And there is no data as such saying that macrophages in human express arginase, but neutrophils do express arginase. And we also have seen that the paper citing that during such chronic inflammatory diseases, arginase is upregulated in humans as well, but we are not sure which cells.
Okay. So that remains to be determined. Potential disease in which polyamines are important is Leishmania, because it grows on the polyamines.
Mm-hmm.
And you also mentioned that, I think in your introduction, you mentioned that two mouse strains that differ in a transporter for arginine show differences in susceptibility to Leishmania on page 16, I think it is. So are there opportunities for treatment of disease, for treatment of Leishmania? In targeting this system? What do you think? Because the bug so much depends on the polyamines that are produced?
Yes, but in many cases of infection, it is like we are not really able to find what sort of macrophages are really involved, like M1 and M2, though it is a textbook classification when it comes to practice. It is like continuous spectrum of macrophages. It could either be M1 or in between stage or M2. And with some infections with Leishmania, sometimes with some infections we also see that they are stimulated by other cytokines and they are not STAT6 dependent and all that. So, and if it is from, if you would like to know about the polyamine, then again it has to be catalyzed by arginase enzyme, and I am not sure like how far this would work.
Okay, so you would first need to generate data on what cell types are exactly involved in this.
Because there are cases in which they exhibit M2 type of surface markers, or they worsen the disease, but then if you collect the wound fluid or something, then you don't find any M2 macrophages, whereas, and they are not STAT6, which is characteristic for M2, so it's like...
Okay, first better characterization. I'm looking at the prorector, whether I can still continue, or should I? Okay, I will give the word back to the prorector.
Thank you, Dr. De Winter.